Transport across the cell membrane. Substances can move into and out of cells in various ways. Some methods occur passively without energy, while others are active and require energy expenditure. A. Simple diffusion. Simple diffusion is the net movement of dissolved particles down their concentration gradients from a region of higher concentration to a region of lower concentration. This is a passive process that requires no external source of energy. 1. Osmosis. Osmosis is the simple diffusion of water from a region of lower solute concentration to a region of higher solute concentration. When the cytoplasm of a cell has a lower concentration of non-penetrating solutes than the extracellular medium, the medium is said to be hypertonic to the cell, and water will flow out of the cell. This process, also called plasmolysis, will cause the cell to shrivel. If the cellular environment is less concentrated than the cytoplasm of the cell, the extracellular medium is said to be hypotonic, and water will flow into the cell, causing it to swell and lyse. For example, red blood cells will burst if placed in distilled water. Freshwater protozoa have a contractile vacuole to pump out excess water and prevent bursting. Facilitated diffusion. Facilitated diffusion, or passive transport, is the net movement of dissolved particles down their concentration gradient through special channels or via carrier proteins in the cell membrane. This process, like simple diffusion, does not require energy. Active transport. Active transport is the net movement of dissolved particles against their concentration gradient with the help of transport proteins. Unlike diffusion, active transport requires energy. Circulation. Circulation is the transportation of material within cells and throughout the body of a multicellular organism. A. Intracellular circulation. Materials move about within a cell in a number of ways. 1. Brownian movement. The movement of particles due to the kinetic energy spreads small, suspended particles throughout the cytoplasm of the cell. 2. Cyclosis or streaming. This is the circular motion of cytoplasm around the cell, which transports molecules. 3. Endoplasmic reticulum. This provides channels throughout the cytoplasm and provides a direct continuous passageway from the plasma membrane to the nuclear membrane. B. Extracellular circulation. A number of systems have been devised to deal with the movement of materials on a larger scale throughout the body of an organism. 1. Diffusion. If cells are in direct or close contact with the external environment, diffusion can serve as a sufficient means of transport for food and oxygen from the environment to the cells. In larger or more complex animals, diffusion is important for the transport of materials between cells and the between cells and the interstitial fluid that bathes the cell. 2. Circulatory system. Complex animals whose cells are too far from the external environment to transport the materials by diffusion require a circulatory system. This generally includes vessels to transport fluid and pump to drive the circulation. Enzymes. Enzymes are organic catalysts. A catalyst is any substance that affects the rate of a chemical reaction without itself being changed. Enzymes are crucial to living things because all living systems have the continuous, controlled chemical activity. Enzymes regulate metabolism by spreading up or slowing down certain chemical reactions. They affect the reaction rate by decreasing the activation energy. Enzymes are proteins, and thus thousands of different enzymes can be formed. Many enzymes are conjugated proteins and have a non-protein coenzyme. In these cases, both components must be present for the enzyme to function. A. Enzyme specificity. Enzymes are very selective. They may catalyze only one reaction or one specific class of closely related reactions. The molecule upon which an enzyme acts is called the substrate. There is an area on each enzyme to which the substrate binds called the active site. Two models describe the binding of the enzyme to the substrate. Model 1. Lock and key theory. This theory holds that the spatial structure of an enzyme's active site is exactly complementary to the spatial structure of the substrate. The two fit together like a lock and key. This theory has been largely discounted. Induced fit theory. This more widely accepted theory describes the active site as having flexibility of shape. When the appropriate substrate comes in contact with the active site, the conformation of the active site changes to fit the substrate.